The human body is made up of billions of cells. These cells form tissues that, in turn, combine to form organs. For all the organs in the human body to form and perform their functions properly, a number of processes need to run smoothly, using the instructions found in our genetic makeup. An individual's genetic material, known as the genome, is made up of DNA stored in the nuclei of all our cells. Let's try a few culinary metaphors to illustrate the composition and use of this genetic makeup. The human genome contains around 20 to 25,000 genes. They correspond to a collection of recipes that are used for the fabrication of the components that are needed for the proper functioning of our tissues. The genome is therefore like a huge cookbook, and the different tissues are the different restaurants, pizzerias, Chinese restaurants, etc., that are responsible for using the gene recipes from the genome cookbook to ensure that our bodies function properly. The genes are like recipes for making a dish, for example a pizza. The genes are composed of introns and exons. The totality of the genome's exons is like the list of all the ingredients needed to prepare the recipes in the cookbook. The combination of these ingredients is what makes it possible to prepare the many different types of pizzas. This collection of exons is called the exome. Introns don't provide any additional information, but they do contribute, from the same recipe, to the combination of different ingredients and thus to the making of several different pizzas. For example, the Regina pizza with tomato, cheese, oregano, ham and mushrooms, or the Margherita pizza with tomato, cheese and oregano. Different combinations that our tissues require to function properly. Although the exome accounts for only 1 to 2% of the genome, it is in the exome that we find the vast majority of anomalies known as variants or mutations, which are responsible for rare genetic diseases. A variation can, for example, make an exon disappear. Thus, an ingredient such as ham or mushrooms is no longer available, which means that the Regina pizza can no longer be made in our restaurants, the tissues that make up our organs. The remaining 98 to 99% of the genome, the introns and the regions that separate our genes, are called non-coding regions. These non-coding parts of the genome play an essential yet little-known role in regulating gene expression. In other words, the use or not of the different recipes. In fact, depending on the tissues, always equated to our restaurants, and depending on the body's developmental needs, lighter summer recipes or heartier winter recipes may or may not be used by the chef. Previously, this non-coding genome was considered to be of no interest and was referred to as junk DNA. But that's not the case. For many years, the tools used to identify the cause of rare diseases were not very effective. In the early 2010s, thanks to the development of new high-throughput sequencing technologies, a veritable revolution took place. Next-generation exome or genome sequencing makes it possible to find the cause of the disease, the responsible variant, in 50% of people with rare diseases. For the remaining 50%, carriers of a rare disease with no known diagnosis, how can this impasse be resolved? To return to our culinary metaphor, why is the recipe for a Regina pizza no longer used or served in a restaurant when that restaurant is Italian, and the Regina pizza appears on the menu as usual? This lack of diagnosis may be due to technical or knowledge limitations. Also known as variants of uncertain significance, or variants in non-coding parts, which are still poorly understood. They are still difficult to identify or to implicate in the disease. To take things a step further, the combined or sequential use of new techniques could help to identify a diagnosis. This is what we call omics, or even multi-omics. With this approach in mind, let's take a look at how we can move forward in the current state of knowledge. If necessary, to ensure that the instructions on the DNA molecule are available to the cell, these instructions must first be copied into molecules known as messengers or RNA transcripts. These are photocopies of recipes that can then be used in the kitchen. The collection of transcripts from active genes can vary according to the cell or tissue type. This collection is called the transcriptome. Each tissue or cell, similar to a restaurant, uses only a subset of the recipes listed in the cookbook. What's more, the transcriptional process for the same gene can vary from cell to cell and over time. It allows the use of a different combination of exons per gene, and so of ingredients from the list. This process is called alternative splicing. It defines variations of the same recipe with, for example, not using a particular ingredient, such as the absence of ham on the Regina pizza when it is served in a vegetarian restaurant. Transcriptome analysis refers to the techniques used to analyze RNAs. That's to say, copies of the recipes used in the kitchen and for the level of expression of genes in a tissue. It is a test designed to determine whether a recipe available in the kitchen is incorrect or used too often, for example. Transcriptome analysis can be used to confirm the involvement of a variant of uncertain significance when it is at the origin of altered expression or a different composition of a gene transcript. Transcriptome analysis can also be used to target a variant present in the non-coding parts of a gene. It is usually performed using RNAs extracted from a blood sample or from skin cells. Transcriptome analysis is always more informative when it is done using affected tissue, if it is available, muscle tissue in the case of myopathy, for example. Each of our cells contains our entire genetic heritage. However, while all of our cells contain the same information, they obviously don't use this information in the same way. A skin cell is nothing like a neuron, and a liver cell doesn't have the same functions as a heart cell. What's more, twins who share the same genome are never perfectly identical, like a Regina pizza from one restaurant to another. In these examples, the explanation for this particularity is called epigenetics. As we saw earlier, and under certain conditions, some recipes are not used because they are hidden. In a restaurant, the menu can change according to the weather. For example, hot, hearty soups are out of fashion in summer. The study of the epigenome corresponds to the study of changes in access to the different recipes. Even though they do not involve variations in the DNA sequence, these changes can be transmitted when the cell divides. As opposed to variations affecting the DNA sequence, the very text of the recipes in the book, epigenetic modifications can be reversible. The epigenome thus consists of marks on the genome, like pieces of repositionable adhesive tape that will cover or uncover recipes to indicate when and where the recipe will be accessible or not. Epigenetics is focused on an additional layer of information that defines how these genes will be used or not by a cell. A gene is then said to be active, switched on, or inactive, switched off. If these epigenetic modifications persist, even when the signal that induced them disappears, this can lead to a genetic disease. The study of the epigenome, in other words, the analysis of the distribution of these marks throughout the genome, can reveal genetic diseases for which there is no DNA modification, but rather there are epigenetic modifications. The epigenome can also reveal genetic diseases that involve DNA modification, which results in a characteristic epigenetic profile. But it's not only genetic factors that cause epigenetic modifications. The environment, such as diet, smoking or stress, can also lead to epigenetic modifications, that is, the switching off of genes. In the near future, other examinations are likely to contribute to our understanding of rare diseases that have yet to be elucidated. the ones for which we do not yet know the recipe, the gene that is lacking. These examinations include proteome and metabolome analyses. The proteome is made up of all the proteins produced in our body by the 20 to 25,000 genes. The same gene can lead to the synthesis of different proteins, which can vary according to the cell or tissue type. In the kitchen of a restaurant, as we imagined previously, the protein would be the dish we eat. Proteomics studies the composition of proteins in a tissue. One gene can give rise to several proteins, thanks to what is known as alternative splicing, a process that makes it possible to use different combinations of exons from a single genomic sequence. In addition to alternative splicing, proteins can undergo numerous post-translational modifications, such as the addition of a methyl or phosphate group. The proteome therefore contains a much larger number of proteins than the genome contains genes. These proteins are the result of all the recipes for the available dishes on the menus of our restaurants, which are our cells and tissues. The dishes are presented with their various ingredients in varying quantities and with varying degrees of seasoning and spice. At present, proteomic analysis is an emerging technique in the field of rare diseases. In combination with transcriptomics, proteomic analysis could contribute to obtaining a diagnosis in cases where a genetic or epigenetic variant is at the origin of an altered production of protein number or quality. The complete collection of all the small molecules, known as metabolites, in a biological sample is called the metabolome. The metabolome includes amino acids, organic acids, sugars, and fatty acids. It reflects the activity of underlying proteins and the state of our body's cells and tissues. The range of metabolites can vary according to cell and tissue type. The study of these molecules, known as metabolomics, involves looking for ingredients in abnormal quantities. In the kitchen, this means deducing, for example, that a recipe in one of these restaurants, the tissues, has been poorly prepared by the chef. Pathological DNA variations in certain genes can result in a specific metabolomic signature detectable in certain biological fluids and tissues. These analyses could thus contribute to the diagnosis of certain rare diseases, much in the same way as proteomics. In recent years, advances in technology and bioinformatics have enabled the development of so-called omics techniques. The term refers to the implementation of a battery of analysis of the content of living organisms at the molecular level. In this video, we have presented genomics for DNA, transcriptomics for RNA molecules corresponding to copies of genes, epigenomics for epigenetic marks on the genome, proteomics for the study of the protein repertoire in a tissue, and metabolomics, the observation of metabolites in certain biological fluids and tissues. Multi-omics analyses make it possible to combine the data obtained through the various omics. Bioinformatics is indispensable for the analysis and integration of the huge amounts of data, big data. As a whole, omics represent a real hope for resolving diagnostic impasses. Multi-omics deciphers the complex molecular mechanics and maps the most elusive anomalies at the root of genetic diseases. The 20 to 25,000 recipes in the human genome are first copied as messenger RNAs in the nucleus of each cell, before being decoded into proteins in the cytoplasm in varying combinations in the muscles, heart, or brain. Proteins determine how the metabolism, in other words, all the chemical reactions inside a living being, occurs, and which products, amino acids, lipids, sugars, will be synthesized, broken down, or degraded. All these processes can be regulated to determine where, when, and how genes are expressed, where, when, and how the 20 to 25,000 recipes are used in the restaurants that are our cells and tissues.